The topic of my talk today is the phylogeny of the cerebral venous system. There's no COI for my presentation. Why phylogeny is necessary? Maybe phylogeny helps to understand the human cerebral venous system, especially venous variations, and it also has some implications for translational research. However, there is not so many papers describing phylogeny of the venous system. These three papers from Phylogeny group published in 2007 and 08, very excellent papers. It's better to read these three papers for further understanding of the venous phylogeny. This is a famous illustration by Ernst Haeckel, the German anatomist, published in 1874. He said, ontogeny recapitulates phylogeny. Maybe scientifically this is not true, but we know in early embryogenesis, even fish or humans has all the similar morphology. This holds true even for the vascular system. This is the early embryologic stage of Medaka, the teleost, the small fish famous in Japan, and the black shows the arterial system, and the gray shows venous system. And the venous flow from the hemispheres go back to primitive head sinus, and which was connected to anterior cardinal vein. Anterior and posterior cardinal vein connect to form common cardinal vein and back to heart. This primitive structure holds not only for fish but for humans as well in the early embryogenesis. This is the phylogenetic tree. If you focus on the terrestrial structures, you will find the large neoparium and neostriatum in the birds and mammals. And also, if you focus on the olfactory bulbs or larger archiparium or the parium found in fish, amphibious, and reptiles. So in my presentation, I'd like to divide the vertebrate into two groups, lower vertebrate and higher vertebrate. In the higher vertebrate, mammals, including the humans, there are mammals, and also I'd like to divide higher mammals and lower mammals. And domestic animal locates in between these mammals. If you look at hemispheres and neoparium, you'll find a large neoparium in the human and also birds. But in a fish, amphibious, reptile, that is lower vertebrate, the neoparium is located in between archiparium and paraoparium, and they occupy very small parts. So the quite different parial structure between the lower and higher vertebrate. If we focus on the environment and leptomeningeal development of the vertebrate, of course, fishes are living in the aquatic environment, and ammonates living in the terrestrial environment. The amphibians are in between, and they are becoming in the transitional period, that is about 340 million years ago. Let's see the structure, the leptomeningeal structure of these animals. In the fish, there are single leptomeninges, and amphibians and reptiles, they have dual or two layers. And only birds and mammals, we have three leptomeninges, a triple leptomeninges. Only birds and mammals are allowed to have dual venous structures. This is an illustration of the leptomeninges of fish and amphibious lower vertebrates. From ventricles, it drains to the dorsal median vein, and also parenchyma drains to the dorsal median vein. This is not the superior saccus sinus. This is a cortical vein running in the subarachnoid space. And interestingly, this is homologous to the median vein of prosencephalon of Markovsky in the humans. This is also illustration of the human, birds and mammals. There are triple left main structures, and from the ventricles drainage to the great vein of Aaron, and then to the superior sagittal sinus. And the cortical parenchymal structures also drain to the dorsal sagittal sinuses. So Pierre Lacha's group proposed four venous drainage system, that is, dorsal, lateral, and ventral or ventricular. And dorsal ventricular venous system related to the neoparium. So it's the superior sagittal sinus, inferior sagittal sinus, sinus, fasting sinus, transverse sinus belong to this dorsal system. Ventricular system is drainage from the ventricle, so then it's quite strongly related to the median proximal veins. From lateral surface of the hemisphere, it's drained through the tentorial sinus. It's a future middle cerebral vein related to the perioparium. And ventricular venous system belongs to the basal vein of Rosenthal, and this is related to the archiparium in the humans. And this is the infratentorial structures, and the dorsal venous system, it's related to the neocerebellum and transverse sinus, occipital sinus, and the marginal sinus belongs to this system. And this is the median vein of proximal. It was found in the early embryogenesis of the humans between 6 or 11 weeks, and then it regressed. Let's see the embryology of the median vein of proximal. Median vein regress, and we call it cranial regression, and fasting sinus regress, and internal cerebral vein. It was connected to the great vein barrier. This is a normal development of a deep venous system. And in the lower vertebrates, they have no superior sagittal sinus. All the veins dorsally drain to the mid-dorsal cortical veins. It's an equivalent or embryological homologous to the median proximal vein. So we may call this the primitive dorsal venous system for lower vertebrate. But in the higher vertebrate, we use dorsal sinuses, so we can call the advanced dorsal venous system for higher vertebrate. Let's see the evolution of the tentorium of the falx. Without falx cerebri, superior sagittal sinus and inferior sinus does not exist. Also, without tentorium cerebri, transverse sinus does not exist. So, the transverse sinus is only found in birds and mammals, and the storage sinus is only found in mammals. Let's go back to the embryology of the cerebral venous system. This is common to all vertebrates at the beginning of the embryology. The initial stage without venous system, before the circulation begins, the brain is maintained by diffusion through CSF. Before the closure of the neural tube is diffusion from the amniotic fluid. After the closure of the neural tube, it is also coming from the nutrition by diffusion from the surrounding meninx primitiva. And then the further increase of the parenchyma, choroidal plexus herniated into the ventricles, and at this stage, 6 to 11 weeks, nutrition was also done by diffusions. At the choroidal stage, you can see the meninx primitiva, then the surface of the embryo, and if you see inside the ventricles, larger choroidal plexus was noted. And the primitive head sinus plays an important role after brain circulation starts through the primitive arteries and veins. This is, you can see the anterior and medial procedural plexus was drained to the primitive head sinus. This primitive early venous system is common to all vertebrates. Then then only the lower vertebrates, they join to the mid-dorsal veins. This is not a sinus. Those are midline veins, homologous to the median vein of protoporphyrin of Markovsky. And those are midline veins connect to the internal jugular veins. This is only hold true for the lower vertebrate. How about the higher vertebrates? Those are dural venous plexus develop from middle or posterior dural plexus, and it plays a major draining role of the brain and prevent head sinus regresses because of the enlarging the trigeminal ganglion and the otic vesicles. So the cavernous sinus is isolated, and in the dorsal portion, there is a prominent venous dural structure develops. This is only true for birds and mammals. And finally, there's an outflow variation developed in mammals. For the higher mammals, and they are drained mainly in the internal jugular vein. For the lower mammals, they use mainly external jugular vein. Only the man uses the internal jugular veins. So I'd like to illustrate this, the lower and higher vertebrate venous system. First, we start with the lower vertebrates. You can see the mid-dorsal veins. This is a subarachnoid vein collecting the venous flow dorsally. This is the hagfish, the example of the fish. And the dorsal view of the venous system, the olfactory veins are collected in the mid-dorsal veins, and then drain to the internal jugular veins. When you see the mid-sagittal section, and this is the venous flow was collected dorsally to the sagittal veins. Archipariums is collected dorsally in the fish. If you see these pictures and illustration, how similar between the structure of the veins and also median vein of protoporphyrin in the early phase of the human venous system, and the dorsally are formed. It is a tiger salamander, the venous system. You will see not the midline large veins, but you can see the venous structures collected to nodus vasculosus and also sacs and lymphatic sac. This is the collector of the venous system dorsally. If you see on the right side the mid-sagittal section, choroidal veins are collected also dorsally to this venous system. This is the xenopus, the frog, and you can see the dorsal view. Of course, in the similar to the salamanders, venous flow was collected dorsally, and in this xenopus, the venous flow was either drained to the spinal veins or internal cerebral veins. This is a turtle, and the dorsal view of the venous system, and large mid-dorsal vein was collecting the hemispheric venous flow. Then further, they drain to either anastomotic veins or directly to the internal jugular vein. Also, this anastomotic vein is finally drained to the internal jugular vein. This is lateral view of the venous system of the turtles. And interestingly, this anastomotic vein runs between the 5th and 7th nerves. This is quite similar to the course of the primitive head sinus. Probably this anastomotic vein is a remnant of the primitive head sinus. This is the birth, and both belong to the higher vertebrate. This lateral view, this large hemisphere and the large midbrain, especially birds has a a large superior curriculum, and the main venous flow through the mid-dorsal sinus and occipital sinus, and then to the vertebral veins, or the temporal rate collecting through the transverse sinus and then to the external jugular veins. And in this birth, the internal jugular vein is very small. This is a dorsal view of the birth. You find the mid-dorsal vein that's running to occipital sinus and then to the vertebral veins. This is rat. And the lateral view, you will see SSS, the superior sinus, and the confluence connected to the transverse sinus. And transverse TS, transverse connects to sigmoid sinus, small sigmoid sinus, and also PSS, petrosquamous sinus. In the rat, the main flow out is through the petrosquamous sinus to the external jugular vein. This is my family members, dogs, 3 dogs, and the view from the buttocks. Interestingly, the flow in the cortical direction of the blood flow in the cerebral veins parallel to that of the dorsal sinus. This is the opposite to the humans. They are same flow direction to the superior flow in the superior sinus or the cortical veins. This is caused because in the humans, rapidly enlarging occipital and temporal lobes cause some distortion of the cerebrum. So initially, the cortical vein were perpendicular to the superior sinus, and ICV and cell sinus are causing in the straight fashion. But due to this rapid growth of the teres balloon, the cortical vein shows retrograde course, and deep venous system, the vein of Galen or internal cerebral vein, show snake course. This is the lateral surface of the dog's brain. No superficial MCV are like human ones, because the ventricular cerebral veins, ventral cerebral veins are anastomosed with basal vein. This is the buttocks view of the veins. Ventral cerebral vein, equivalent to human MCV, was connected to the dorsal petrosal sinus, which is a tentorial sinus. And interestingly, there is no communication between the cavernous sinus and MCV. This is also drainage venous system of the dog, and cavernous sinus has no connection of the no contribution to the cerebral drainage, and those are petrosal sinus are connected to the transverse sinus. And finally, there are 3 possible drainage routes: vertebral vein, internal jugular vein, and external jugular vein through retroglenoid veins. But mainly, their flow is either vertebral vein or retroglenoid venous flow. And the internal jugular vein, the contribution is very small. It's interesting, the MR venography of the dog at 1.5 Tesla. On the right side, you will show the lateral view of the MRV. And the stress sinus connected to the transverse sinus. And then 2 ways, temporal sinus through the postglenoid foramen to the external jugular vein, and or small sigmoid sinus, and then to the internal jugular vein. In this dog's MR, you will find the main flow of the external jugular veins. How about the monkey? This is a lizard monkey, and also cavernous sinus has no communication with the MCV Sylvian veins, and red arrows show this MCV connected to the superior petrosal sinus. And then they have also 2 ways drainage to petrosquamous sinus in the green, and also jugular vein, internal jugular vein, so that it's blue lines. In the early embryonic stage of the humans, the transverse sinus have two possible directions: the sigmoid sinus or petrosquamous sinus. But in human selected the sigmoid sinus, and then the petrosquamous sinus regress spontaneously. This is a general illustration of the dorsal venous system. And dorsal venous system is a superior sinus, transverse sinus. Then it can go to the external jugular or internal jugular or vertebral venous system through emissary veins. Monotreme is the most primitive mammals. It's very interesting. The drainage from the transverse sinus is to external jugular vein is through the stylomastoid foramen. And this course from cavernous sinus to stylomastoid foramen is probably the remnant of a primitive head sinus. So this is a dorsal venous system for biped or quadruped. Quadruped prefers a petrosquamous sinus drainage, but biped, like us, prefers the internal jugular drainage. Let's see the dorsal venous system of the higher mammals. And man, rhesus monkey, and pig cats mainly drain the internal jugular veins. However, the dog, sheep, rabbit, and ox, and the horse are external jugular predominance. But interestingly, only the man uses solely internal jugular vein. And review the lateral venous system for lower vertebrate. The venous fluid collected dorsally to the dorsal sagittal vein. This is homologous to the median vein or prosencephalon. And the lateral venous system, the higher vertebrae, there are several possibilities for drainage. They can go to the transverse, they can go to the pterygoid plexus, they can go to the paracavernous, they can go to the cavernous sinus. But a cavernous sinus that capture is only found in man. It is interestingly, cavernous capture from the Sylvian vein, also medial superior petrosinus found only in man. And this is a search of higher vertebrates, and only human can correct the venous flow to the cavernous. It is interesting to notice a unique cerebral drain in the man. And sigmoid sinus, only the sigmoid sinus drainage are found in only men, and the cavernous capture of the MCV and also medial superior petrosinus found in man. And also, this is interesting in the limbic arch found in the higher vertebrates, because archipalium in the higher vertebrae pushed medially or ventrally. Only this limbic venous arch remains. So anterior and posterior pericallosal veins, and also anterior cerebral vein and inferior ventricular vein provide this limbic venous arch. And this limbic venous arch can be found in the case of, for example, the corpus callosum AVM or IVF. As for the ventral venous system, the higher vertebrate, the primitive tentorial sinus is drained distally to the tentorial sinus, but it's occluded because of the enlarging the hemispheres. Then instead of these superficial veins, basal vein and the ventral system develops. And basal vein Rosenthal corrects the venous flow of transferon, diencephalon, and mesencephalon. We may call it first and second, third segment. Interestingly, third segment may drainage to the vein ganglion, torcular, or transverse sinus or superior petrosinus. It's depend on the the phylogenic ascent. So the most advanced form is drainage to the vein of Galen. Finally, talking on the ventricular venous system. Ventricular venous system is only found in the higher vertebrates. And because the structure, like basal ganglia, red nucleus, crus cerebri, or pontine tegment, was developed quite late. So only the venous system developed in the higher vertebrates. You will find this this ventral venous system connected to the lateral venous system, and they locate the basal vein and the lateral mesencephalic, lateral pontine, lateral mesencephalic veins longitudinally. This review again. The ventricular venous system is a venous system of the forerunner of the median vein of prosperinum. In the lower vertebrate, perineum is drained by this mid-dorsal vein running in the subarachnoid space, but in the higher vertebrate, it drains deep venous system. Dorsal venous system drains the perineum or neocerebellum, and it's only found in the higher vertebrate. It drains the internal jugular vein or through cerebral sinus and or external jugular vein through the petrosquamous sinus. Lateral venous system includes the medial cerebral vein, inferior ventricular vein, or lateral mesencephalic pontine medullary veins. And lower vertebrates, superior cortical vein drains the mid-dorsal veins. In higher vertebrate, primitive tentorial sinus drain ventrally. The ventral venous system is related to the new structure, and this is not found in the lower vertebrate. And conclusion, phylogeny of the venous system was presented. This knowledge is important to understand the cerebral venous anatomy and variations, and this possibly contribute to the safer treatment. Thank you for your attention.